Hello everyone, and thank you for joining us today as we talk to the scientist. My name is Dalton Dietrich. I'm the Scientific Director of the Miami Project, a Center of Excellence at the University of Miami Miller School of Medicine. Today I'm speaking with Dr. Dan Liebl. Dan is a professor of the Department of Neurological Surgery and a very important member of the Miami Project. Dan, thanks for joining us today.
Thank you.
Dan, how many years have you been with the Miami Project?
Pretty much my whole faculty career, so going on 20 years now.
So Dan was recruited out of an outstanding postdoctoral fellowship to the Miami Project as assistant professor, and now has made it all the way up the ranks to full professor. And Dan is what I consider a modern scientist. He's working on both basic and translational work. So Dan, tell us a little bit about the challenges of the scientists in today's, with the high technology and different disciplines we have to deal with and incorporate into our science.
Okay. When I first started, science was very different than what it is today. In fact, science is kind of an evolving industry. It's one of the most evolving industries that we know of. We have knowledge that's produced worldwide, and we have to incorporate all that knowledge into how we define our questions. And the technologies that have advanced over the last 10 years have really excelled how we look at science in general. So many years ago, scientists could either be really good at looking at a specific protein or molecule, or they could look at a cellular function, and they were experts in their field. And when they brought a lot of different scientists with a lot of different backgrounds together, they could understand a fundamental question. For example, when we first understood how neurons communicate between each other, there's people that looked at the electrical properties, electrophysiologists. They looked at the chemical properties, such as cellular biologists and molecular biologists. But not any one of those scientists necessarily looked at it in a comprehensive way. Today's scientists really need to look at it in a very holistic way. They need to use a plethora of different outcome measures. So they need to use everything from looking at genes to whole animals and using various techniques. But they also need to look at it in a way that allows them to ask questions based upon our technologies. So some of the advancements have been that we can look at the outcomes of one event, such as adding a drug, mutating a gene, and we can look at how the whole cell changes. We can look at gene-wide changes, we can look at protein-wide changes. And these changes give us a better picture of what's happening. And why that is important is basically that when we think of translating something from the benchtop to the clinic, we have to think about what's going on within the whole system. And in fact, when we look at the number of clinical trials that have been successful, there are very few. Most trials fail. And why? Well, there's a lot of different reasons why they fail. But one of the reasons is we really don't have a big picture idea of what's happening when we add a drug, for example. So our technology today are allowing us to do better science, to do more sound science. And to be able to do that, we need to understand how to utilize those technologies within all our own laboratories.
Very good, very interesting. Yes, we have a lot of challenges today, and being in the Miami Project offers some advantages, right, in terms of the complexity of the science now and having to be able to interact with different colleagues within the project and throughout the University of Miami, and in fact throughout the world in today's ability to do that efficiently. So can you tell us a little bit about collaboration?
Yeah, so collaborations is essential. We can't do everything, and we're not experts at everything. So our ability to do a fundamental project that we understand what the components need to be and collaborate with those professionals around the world that would best fit that project is essential to success. And we have established, like you said, collaborations within the Miami Project as well as collaborations outside the Miami Project. And some of the best collaborations we have are some of the neighbors that sit around us, because they think of questions in very different ways than we do. But when you're right next to someone, you can actually feed off each other, and you can do better science, and you can use tools that they may have that you don't have, and vice versa. Our collaborative unit within the Miami Project is essential, and I think it's one of the strengths of the Miami Project.
And if you pick up one of our publications, you'll see many times we have 4, 8 many types of colleagues on these papers that are contributing significantly to the science. And with that, we can utilize different types of cutting-edge research that we may not be, the primary pusher of that particular or motivator of that particular technology, but we can initiate that technology into our scientific question. So how does your scientific programs fit into the MAMI Project goals, Dan?
Well, I think they fit in well.
I agree.
So one of the ways, so how do you define fitting in? I think fitting in to me means how do we push the boundaries? How do we push the limits? How do we take our science and look at things in ways that have never been looked at within the project? And then how do we advance not only our own program, but everyone else's by doing so? And I think our science does it in many ways. One, like many people in the MAMI project, we have a very benchtop to bedside approach to science. So we want to do discovery science. We want to be able to understand how things work, the mechanisms that underlie our projects, but also how to translate those to the clinic. And preclinical trials are something that the MAMI project is a strength in. And I think all our different programs come together to help advance those strengths.
So yes, to just emphasize that point, as scientists come in and they can work on molecules, they can work on genes, proteins, but at the end of the day what we're trying to do is take that discovery knowledge and move it into translational models and ultimately move it into people to actually change medicine now.
One of the things that is really an advantage to the MAMI project too is that we're in the Department of Neurological Surgery. So there are neurosurgeons that work here, and in recent years we have become more collaborative or work together with some of the neurosurgeons and allow us to actually test things not only in human patients as we do in preclinical and clinical trials, but also be able to understand how our questions can be reproducible in humans. So there are human tissues that we can utilize, and there's different colleagues that we work with to obtain those tissues to be able to advance our science and make sure they are going to be successful in humans.
So we're sitting in Dan's laboratory now, and it's very active. So Dan, maybe you could tell us a couple about really exciting projects you're working on with your colleagues now at the moment.
So I think all the projects in my lab are exciting, or else we wouldn't be doing them. But if I had to focus on a couple, one of the projects that we're really, it's kind of a new project, and it really kind of attests to how we think about developing new projects in the lab, is to look at how cells are maintained after injury. A lot of people look at specific cell functions, specific, like I said, proteins and signaling events, but when we think about injury, we think about two things. We think about how to protect neurons or cells after injury and how to restore or regenerate those cells. And these questions are not completely exclusive of each other. One of the projects is to look at how we stabilize the connections between cells. And we know after injury, many of these connections are destabilized and they're lost. And some of the behavioral deficits that we have, whether it's motor deficits, cognitive deficits, and others, are resulting from the disconnection between these cells. So we are really investigating how the cell actually forms these connections, how they stabilize these connections, and some of those questions are used in how we regenerate these connections after injury. Another area is to look at a concept that isn't relatively new. It's to look at how cell membranes function. So we've, back in 100 years ago, we understand that membranes are essential to cells. They form the units that surround the cell. They hold everything in. All the signaling is dependent upon membrane function. And the membrane's made up of a lot of lipids. So of those, about 90% of those lipids is cholesterol. So how is cholesterol regulated after injury is really a key question. When you go into the literature, you see that there's very few studies that have actually looked at this. In fact, most of our information about cholesterol regulation comes from genetic disorders, such as a rare disorder such as Niemann-Pick's disease, where there's a cholesterol storage defect. Well, in the injured brain, we don't deliver cholesterol properly to the membrane either. So some of our group in our laboratory are investigating the factors that regulate cholesterol trafficking within the cell with the hope that we can actually develop drugs to help protect or lead to homeostatic cholesterol levels and a better functioning cells. And what will that do? Well, that will prevent cells from dying, will promote regeneration, and all the cellular functions that go along with it.
Dan, in addition to being an outstanding scientist, you're also a mentor and educator. So let's talk about a little bit about how important that is in terms of training the next generation scientists on the science that we're doing in the MAMI project.
Well, education is fundamental for science to succeed into the future. If we don't educate the next level or the next generation of scientists, we won't have a next generation of scientists. And the world is challenging these days with the number of scientists that succeed in their position, the number of positions that are available, but to train the very best is critical. And there's many different ways that we can train them, but going back to the fundamentals, training them to be a sound scientist is important. And I've worked at the university, I've mentored many students, I've been the director of the neuroscience program for several years. I also now work with physician scientist students and training those individuals as well. And ultimately, I think the success of the University of Miami is dependent upon training science in general and training graduate students as well.
Very good. Thank you very much. Well, this has been a lot of fun. Thank you, Dan, for visiting with us briefly today, and we'll have you back when other exciting research in your lab moves forward, and hopefully with your help we can make a big difference in people's lives. Thank you so much.
Thank you.